The presence of the Romaine hydropower complex changes the river's flow regime. Hydro-Québec is committed to conduct environmental follow-ups until 2040 to monitor changes in the environment and to assess the effectiveness of mitigation and compensation measures. The follow-up includes a study of primary production.
Primary production refers to organisms living in the water, plants called phytoplankton and tiny animals called zooplankton. These organisms drift in the water. They rise and fall with the tides according to the currents. Many of these organisms can't be seen with the naked eye, yet they're a vital part of the food chain. Tiny, near-invisible phytoplankton and barely visible zooplankton are the basis of the food chain, which leads right up to whales. So it's very important to monitor primary production since it determines all production in the Romaine estuary. The study on primary production has two objectives. The first is to monitor habitat quality, everything that affects the water column in which these organisms live. We monitor the temperature, salinity, tides, and currents of the waters carried by the river. Our second goal in monitoring primary production is to assess the health of the zooplankton and phytoplankton drifting in the Bay of the Romaine. We monitor plankton up to the Canal de Manguin to assess the impact of the impoundment of the Romaine complex. This study covers a much wider area than our other studies. That is because primary production can be found much further offshore. So we monitor the mouth of the Romaine and the entire section around the Manguin Islands, far into the St. Lawrence. We want to record all biological productivity in the area. To monitor the state of primary production, we look at what's happening in the water. We measure the temperature, salinity, and tides using various instruments. We've also installed instruments on buoys to collect data over about 4 months, from early May to mid-October. We take readings regularly, every 20 minutes. We also check nutrient levels in the water. Nutrient levels determine how much or how little biological production there will be in the area. To track biological production, we measure chlorophyll. It shows how much zooplankton and phytoplankton is in the water, especially phytoplankton. We monitor these levels by moving instruments around the bay or by installing them on buoys in the open water. We'll also assess plankton health by examining species in the water. The quantity and types of species are key health indicators. We use a vertical net to collect organisms in a given amount of water. Then we identify them to determine the community structure that's present in the water. To see what's going on with the community structure, we examine the samples. Since a lot of these organisms are microscopic, we have to examine them with a binocular microscope, a large magnifying glass used in laboratories. The phytoplankton is identified at the Université du Québec à Montréal, and the zooplankton is identified at the Université de Montréal. In addition to tracking the parameters that affect the water in which these organisms live, we track other parameters using our instruments on the buoys and other types of instruments all across the bay and the mouth of the Romaine. We often use these instruments to create profiles. We'll measure the temperature, say, at the surface down to the bottom, or the salinity, for instance. We'll also take samples at specific depths to measure chlorophyll levels, suspended particles, nutrient salts, and other parameters. We collect samples with bottles placed at different depths. We call them Niskin bottles. They're tied to a cable and dropped to a specific depth. We send a weight, called a messenger, down the cable. The messenger hits the bottle and closes both ends, trapping the water inside before it's pulled back up to the surface. The water collected at different depths is sent to a lab, where it is filtered in a filtration column. The filter collects matter in the water, suspended particles, chlorophyll, phytoplankton, and zooplankton. The filtered material is then analyzed at the Institut des Sciences de la Mer in Rimouski. Based on these results, we can assess the health of the Bay of the Romaine and the Canal de Mingan. Our results so far show that the Rivière Romaine is having a minimal impact on primary production in terms of nutrient salts and suspended particles. There is some variation, but nothing out of the ordinary. Our follow-up will allow us to better document any variations. The most important point, as noted in our impact assessment, is that the Romaine contains a small volume of water compared to the immense volume coming from the St. Lawrence. So primary production in the Bay of the Romaine is really influenced by the St. Lawrence.